So today we are having this INBIOSIS-MSBMB hybrid seminar lecture series. This year, this is the first one. And in the past, MSBMB and INBIOSIS have organized a series of seminars. And today, we are very honored to have Professor Fred Antson from University of York visiting INBIOSIS and giving this seminar with a title of Protein Nucleic Acid Interactions, Symmetry Arguments. And I would like to share with you now in the chemistry department of University of York. The Moscow Institute of Physics and Technology in 1986, and earned his PhD from the Institute of Crystallography in Moscow. After working with Keith Wilson at INBIOSIS Hamburg, Germany, he joined Guy
Structural Laboratories Laboratory at York in 1992, YSBL that we know, or we normally call it YSBL. And Fred used or applied electron microscopy and X-ray crystallography to investigate protein nucleic acid machines found in viruses, and with particular focus on DNA packaging models involved in assembly of double-stranded DNA viruses. His research is supported by Wellcome Trust for the past many years. So let's welcome Professor Fred Antson.
Right, thank you very much. It's really great to be here. Shall I use this microphone? This one, okay. Right, so let me try to share my slides. So it's called Disable Participant Screen Sharing. So, yes, so Leon suggested I give you some background about maybe a little bit about where I come from, but also maybe more specifically about the structural levels of our New York. So I'll start with that. To tell you what my group is doing. This focus on natural symmetry and interaction of proteins with nucleic acids. What's my password? Sorry. Okay.
Yes. No. Yeah.
So, okay. This one. Right, thanks for holding me out, and I hope everything works through Zoom as well, right? Yeah. Brilliant. Right, so I'll start from geographic background. So I come from Estonia, which used to be the smallest Soviet republic in Soviet Union at the time when I studied, and I got my education. Sorry? Yeah.
It isn't share in Zoom.
Sorry?
It is share in Zoom.
So I studied in Moscow and got my education in Moscow as well. And just wanted to show sorry for the mess. It's really my fault because I have to bring my laptop. So it's so you need to get the admit.
No, it doesn't matter.
Can you admit from your computer?
Yeah.
Okay, good. Right. Yeah, so I studied in Moscow, and during my PhD did some work also at synchrotron source in Siberia, in Novosibirsk. And then after my PhD, I moved first to Hamburg, to EMPL Hamburg, which was structural biology laboratory, which was headed by Keith Wilson. And then from there, in 1992, I joined York Structural Biology Laboratory. So in Moscow, during my master degree, I did electron microscopy of enzyme called tyrosine phenol lyase. That was in Institute of Crystallography. You know, when I determine structure, shall I explain it like that? When I determine structure as a biologist who were working with this enzyme is actually used for biasing cell Doppler, which is drug used for treatment, major drug used for treatment of Parkinson's disease. And this enzyme is used by Gina Motor Incorporated in Japan for production of L-Dopa. So when biologists ask, so how the structure looks like, and I just say it looks like a block, right? Because electron microscopy at the time was really at a level where you can determine structure only low resolution. So for my PhD, I moved from electron microscopy lab, which was on the ground floor, to the third floor, where we had X-ray structural biology lab. And during my PhD, I determined this detailed X-ray structure of tyrosine phenol lyase. As I mentioned, also this involved building a crystallographic station at the synchrotron site. So this was the first electron storage ring, which was actually built in Siberia, in Novosibirsk, in the Institute of Nuclear Physics. And we actually had to build this crystallographic station from the scratch by studying focusing monochromator and some detector. And our detector was for some reason I can't move my slides forward. Oh, they are actually. So at the time we were collecting data using X-ray film, and this was very laborious because first you collect data, then you develop films in photo laboratories, then you have to scan every film. The dynamic range was very low, and in 1990, a big revolution in X-ray crystallography happened. And this was because at ENBL Hamburg, a new type of detector was developed. Detector which recorded X-ray images, and in our case, images diffraction patterns using image plates. and the readout from this detector was 41. So that was really fantastic. So I moved to EMBL Hamburg in 1990, and in 1992, I joined structural biology laboratory at UOB. So this lab was established in 1976 by Guy and Eleanor Dodson. And the lab became one of the biggest and, arguably one of the most successful structural biology labs in the UK. The lab contributed, to really wide ranging area, not only to structure determination and data analysis, but also development of software, which is now used in every structural biology lab, like programs like COOTS, REFMA, etc. And contributions of INBIOS have been recently recognized even by Green's anniversary prize. This is very rare prize. I think our university, which is like 60 years old, only received 2 such prizes. So one of these came to structural biology lab. So my group is specializing in studying nucleic acid interactions, protein nucleic acid interactions, and try it again. No, my hands are not as good as yours. Oh, you have to move the cursor. All right. So, okay, so maybe, hopefully it will be working now. Yeah, so, and the first, independent project I started was on a protein called TRAP for tryptophan RNA binding attenuation protein. This protein was provided by a collaborator, Paul Golnick, and protein worked by being involved in selecting between 2 different RNA structures. So when this protein was activated, this kind of anti-terminated structure formed when tryptophan operon was transcribed, and this was the lead region of the RNA transcript. And when this structure forms, then the whole operon will be transcribed, and proteins involved in biosynthesis of tryptophan will be produced in the cell. And when this protein is not activated, then this transcription terminator formed, and this leads to transcription termination. So I was provided this protein, and if I say how it looks like, also it's a dimer, because at the time the structures of many transcription factors were already available, and most of them were dimers. Dimers will contain 2 DNA or RNA binding or whatever domain. So when I crystallized this protein and calculated self-rotation function, it immediately became clear that there are peaks which are separated by 32.7 peaks. If you divide 360 degrees by 32.7, the number you get is 1. So I told Paul, can it be 11 cellulose protein? And at the time, this was 1985, no one actually heard about it. He said, "No, Fred, you're crazy, this might be possible." So it was interesting, interesting time. So we had to determine actually structure and show that it was 11 now. Where 11 subunits in this protein, and activating tryptophan molecules actually bind in a cavity between adjacent subunits of the 11 subunits of the gamete molecule. And this will explain how this protein works. So again, the protein is involved in selecting between 2 alternative hairpin structures. So these hairpin structures form after the lead region of the tryptophan operon is transcribed by RNA polymerase. So in the absence of protein, this anti-terminator structure forms, and transcription follows to structural genes. And proteins which synthesize tryptophan will be produced, and then tryptophan will be biosynthetically produced in the cell. However, when there is already enough tryptophan activates this molecule by binding to TRAP. And what actually TRAP does, turned out it binds 11 UAG and GAG triplets. 11 triplets which are separated by 2 or 3 nucleotides. And by doing that, this protein prevents formation of this anti-terminator hairpin and allows formation of the terminator hairpin, which signals RNA polymerase stop transcription. Because again, there is no need to transcribe these genes and no need to cyclophosphamide this binding. This protein is involved in selection between these two structures and essentially regulating transcription. And then I was lucky enough to actually crystallize a complex between this protein and RNA. And Paul Golnick, still not working. Yeah, so hopefully it works. Yeah, so Paul Golnick looked at interaction. So he did some biochemistry and worked out KD for RNA containing different number of repeats of GAG and UAG triplets. And it turns out that strong binding requires at least 6, 7 UAG or GAG triplets. And when you have less than 5 bindings, RNA bindings, a very, very low KD. So there was essentially no binding detected when there were only 2. And then eventually we determined structure of this protein with RNA. This slide nicely shows how RNAs are specifically recognized by this protein. So each of these triplets, which are shown in green here, so that was RNA containing GAG triplets, binds in a pocket where G, A, and so this GAG triplets, each of these nucleotides, bases of each of these 3 nucleotides are making specific interactions with amino acids from the protein. And these 3 bases are separated by 2 nucleotides which do not make any interactions with the protein. This really, the structure shows how the circular assembly generates specificity. Because if you think about nucleic acid or RNA, 3 nucleotides, they don't really provide specificity. It's a very flexible molecule. So you don't need to have having transcription factor which very specifically binds to single GAG triplet would be a very bad idea because it would stick to lots of such GAG triplets present in the RNA. But when there are multiple number of such repeats, this really generates specificity and allows this protein to very, very specifically bind to one such site, only one such site present in the RNA. Okay, so that's kind of a really good example of specificity generated by circular symmetry of the protein. And yeah, so this stage, I was really lucky to get a couple of papers in Nature. Actually, I remember submitting FOSI papers to Nature, and they were all accepted straight away. And I thought, well, it's probably very easy to publish in Nature, right? Then it took me like another 12 years after that to get to Nature. So, yeah, but at this stage, I applied for Wellcome Fellowship. That was 1998, and I was very lucky. I was continuously funded by Wellcome Trust. And currently I have Investigator Award, which is still running until 2027. So I wanted to say a few words about Henry Wellcome, who actually established Wellcome Trust. So Henry Wellcome, he was quite a rich person, but his wife died of unknown disease. and he actually created this foundation. And all of the money he had actually went into this foundation of his which provides funding for biomedical research. So the amount of money which in this foundation is actually much greater than, for example, in the Nobel Prize fund, right? But because everyone knows about Nobel Prize, Alfred Nobel, but not many people know about Henry Wellcome. But Wellcome Trust provides currently about £1 billion every year of funding towards biomedical research. This actually goes not only to UK labs. I think they're very much open to actually research labs also from Malaysia and from other countries. And if you are in London, so this is old Wellcome Trust building. I would really encourage you to go to Wellcome Collection. So there is a museum in the old building of Wellcome Trust, which is open to everyone. So it's free attendance, and it contains lots of artifacts which were collected by Henry Wellcome during his life. Artifacts which are related to human health. There are all sorts of interesting objects, like objects which show that someone performed operation on human skull 400 years ago, somewhere in Central Africa, believe it or not. So yeah. I've been at chemistry department in York for all these years, and with this Wellcome funding, I really concentrated over the last 20 years, on viral nucleic acid machines. So nucleic acid machines which are used by viruses for different purposes, for storage of their genomic RNA or DNA, for translocating viral genome, for several other purposes as well. This is some old slides. So as you know, viruses have been with us, of course, probably as long as human life actually been on our planet. And every year there is another pandemic. So I think when I made this slide, it was like we had just Zika, and this happened, I think, after the Ebola pandemic. Of course, then we had coronavirus. Yesterday, World Health Organization announced that monkeypox virus is a kind of big, emerging problem. So yeah, viruses are cool. Viruses are, of course, interesting. Viruses are very important for human health. Okay. So we looked at a number of different proteins, and one of these was E1 helicase. So this is again circular structure composed of 6 identical subunits. So we determined structure in the presence of ATP, and also we can only see phosphates of ATP. But what was really unusual about this helicase was that even though every subunit was chemically absolutely identical, there was a symmetry in the central tunnel. And it is central tunnel where actually action happens. And when structure was determined in Joshua Torbrook in complexes DNA. So we tried all sorts of very clever experiments to get crystals, but we failed. And they just did very dirty experiments which worked. So sometimes do something quick and dirty, it may work. But anyway, that was a really beautiful structure, which shows that 5 nucleotides of DNA. So this protein unwinds the double-stranded DNA and moves along a single strand of the DNA. So in 5 subunits, there are 5 hairpins, each hairpin interacting with consecutive nucleotides of the DNA, forming a staircase, right? And firing of ATP consecutively around the ring of 6 subunits drives movement of this protein, drives and fuels this protein to move along the single strand of the DNA and unwind double-stranded DNA. this is a really nice case where you have kind of still the symmetric assembly, but it's not really symmetrical, and there is kind of an intrinsic symmetry inside the tunnel, which is important for translocation for the motor to work. And whenever there are circular proteins involved in some sort of motor movement, I think you should really expect a symmetry, I think, and that's what we found in through our work in my lab. Okay, so so we also looked at structures of nucleocapsid proteins, and this work was actually done from Li Sheng, who joined from Yong's lab. So he came to do PhD, and that what shall I do? And I asked, Sheng, what do you want to do? Do whatever you like. And Sheng was like, I'd like to work on Nipah virus. So I said, okay, so he actually got genomic DNA for Nipah virus from Professor Tan, from Kiev, from Kuala Lumpur. Yeah, he was yesterday's meeting. So Sheng produced this protein in E. coli, and actually here is a negative stain picture of the nucleocapsid assembly. So it's forms a long rod. This is a very low resolution picture, and inside this long rod, this rod-shaped helical assembly protects, binds genomic RNA, protects it. And Sheng produced this protein in E. coli, and bound some in E. coli bound some random RNA segments present there, and he then was able to determine structure by cryo-electron microscopy to 3.4x resolution. And in this structure, it became clear that RNA is bound in this groove between two adjacent subunits of between two adjacent domains of one subunit. And there are, I think there are 13 subunits per, turn of this, of the helix of the nucleocapsid. And structurally nicely, showed the mechanism of RNA binding. So protein binds RNA non-specifically, but interestingly it binds in a kind of a C base in, C base out manner, where C bases of 3 consecutive nucleotides are projecting towards the protein, then bases of 3 subsequent nucleotides will be projecting outside. And Sheng was actually very productive. He also produced SARS-CoV-2 nucleocapsid proteins. So I think the first lab in the UK, at least, when the pandemic started, set up this project, got this protein and sent it to all sorts of collaborators. And I think Sheng got a couple of papers out of there. So one was published in JMON Bio. And this paper, this was like 66 sources paper, but it came out in Nature Immunology. Okay, so the main focus of research in my lab is to understand how viral particles assemble and how they fill the capsids with genomic DNA or RNA. And this project I was actually talking yesterday about this, it's a kind of extended talk at the Biochemical Society meeting. So the main project is to really understand what is happening during DNA translocation. And the motor which fills empty capsid with DNA, and this mechanism is actually conserved in bacteriophages and also evolutionarily related herpes viruses, which contain use the same machinery, very similar machinery for filling their capsid with DNA. So the machinery contains C proteins, and the assembly starts from formation of pro-capsid around portal protein, and then another protein, which is called small terminase in the case of bacteriophages, specifically binds DNA, docks it to the large terminase, and then complex of these two proteins dock to portal protein, and DNA translocation in the presence of ATP begins. And when capsid is being, after it has been filled with DNA, one of these proteins will make cut. Some tail of, in the case of bacteriophage tail, which is assembled in a separate pathway, will be attached to the viral head. So the motor contains C proteins, which are shown more or less in scale here. Portal protein, which has central tunnels through which DNA will be translocated. Small terminase, which specifically recognizes viral DNA. And also a large terminase protein, which exists as a single subunit molecule in solution. So we first determine structure of the portal protein. So the protein has 12 subunits. And here is a cross section of this protein. So it has a constricted part, which is only 1.8 nanometers in diameter. And DNA will be brought to the portal protein at this end. So DNA is negatively charged. And as you can see, this part of the tunnel is also negatively charged. So this is presumably important for DNA to be for smooth translocation of DNA, so that it doesn't really bind to the walls of the protein, and because the walls have the same charge, right? So there will be so the DNA. This will help DNA translocation, and when we determine structure of the protein of this protein, because it forms natural DNA pore, we were contacted by Oxford nanopore technology, and we formed this collaboration with Many Vanonov, who was also collaborating with Oxford nanopore technology. But I don't know, do you use ONT sequencing devices here? Yeah, right. As the current device is based on kind of two different proteins, one forming a pore, and another driving DNA into the pore. But they're really interested to look at all sorts of other pores. And because our pore was actually natural DNA pore, not a transmembrane protein, which is used currently by Oxford nanopore technology. So they asked us to see whether this protein can be adapted for and used as a nanopore. So we label this protein with cysteines, and then through molecular conjugation bound porphyrin rings to this cysteine, and that actually shows that it can be docked into lipid bilayer and used for in nanopore technology. And because there are different types of nanopores, and lipid bilayer nanopores, which are currently used by ONT, have their own disadvantages, simply due to the flexibility, not enough a long shelf time. There are efforts currently going into creating so-called hybrid nanopore, where you would actually use solid nanopore, so pore drilled in a solid material, solid state material, and then dock a protein pore which contains the size diameter of the internal tunnel. Trouble with solid pores is that it's very difficult to productively make tunnels exactly the same diameter. So that's why pores which are contained in protein molecules are preferential. And the idea here is to drive protein into solid state pore and then use this hybrid pore for nanopore technology. And we actually show that this pore, so if you do measurements, if you apply voltage, this voltage, because portal protein has negative charge at one end, the positive charge at another end, so it will kind of electrostatically cork portal protein into the spore, and then this voltage can also drive charged molecules through the pore. And here are actually measurements of the current through the pore. So if you add a small protein molecule, you can see the pore becomes blocked, the current drops, and you can measure, this delta I difference in the current, and also the length of the dwell time, length for which the pore has been blocked. And in this case, we did measurements with insulin, DNA hairpin, protein peptide, single-stranded RNA, and you can see that measurements, the delta I versus log TD actually all fall into 4 different areas. I think that the screen didn't actually show up here. So, you can immediately see that you can actually use this for sensing applications, sense what type of molecule is present. So this is something, we are trying to develop further. Okay, and another property of this machine is small terminase. A small terminase again forms a nice symmetrical 9-subunit assembly, and it recognizes DNA in a way similar to the truck protein, which I talked at the beginning, recognizes single-stranded RNA. So in this case, protein interacts with major grooves of the DNA. So every time DNA makes a full turn, one of these N-terminal DNA binding domains would actually slot into the major groove, one of its alpha helices. And in this case, like in the case of TRAP, weak binding of individual DNA binding domain to DNA is compensated by multiple subunits and multiple interactions. Okay, so we also determine structure of the same protein, but from different type of bacteriophages, which package DNA in a somewhat different way. And interestingly enough, even though these viruses are conserved across evolution, it turned out that protein with the same fold can bind DNA inside the central tunnel. So some viruses recognize the viral genomic DNA before packaging it into capsid by wrapping it around. But these particular group of viruses, which are called false viruses, they recognize DNA by binding it inside the tunnel. It turns out that this protein can actually slide along the DNA while sensing what is the sequence of the DNA. And when the sequence is right, when there is the so-called cos side, the flexible N and C terminal segments of 2 adjacent subunits would actually specifically recognize the side, form very specific interactions, and arrest DNA. And this will signal this other motor protein to make cut. So again, this is another example of circular assemblies. The circular assemblies are very versatile. They can bind nucleic acids, both wrapping around outside or using internal tunnel. Okay, so right, and more recently we determined structure of the complete sheet. So here is an empty capsid. So we added DNA. This work was done by Dorothy Hawkins, actually now joined the Crick Institute in London, so she's now a postdoc there. So in this case, we derived structure of this machine from actually capsid. And here is portal protein and also large terminase protein, which interacts with portal. And it turned out that large terminase forms circular assembly of 5 subunits, but this assembly is completely asymmetric. This is a motor protein which drives DNA into the capsid, and each subunit has the major difference between each subunit is that there is different distance between 2 domains of the motor protein, and DNA by such ATPase is usually translocated by Inchworm mechanism. So the protein actually has high sequence similarity to Inchworm monomeric cases. But there are 5 subunits. In this case, it's not monomeric. And we believe that the DNA will be translocated by conformational changes taking place around the circle of these 5 subunits. So by wave-like conformational changes, which are fueled by ATP hydrolysis. This will drive DNA to the capsid. So to summarize, we observed a number of different circular assemblies, and some of these are completely symmetric, and you know, they're quite solid, have circular symmetry. But some of the circular assemblies are symmetric, and these are observed in molecular machines where each subunit is found in a unique conformation. And of course, you know, if you need to bind a very long, flexible polymer like genomic RNAs, then you know, ideal assembly will be helical assembly, like in the case of the NEPA virus nucleocapsid protein. So I'll now try to, I don't know how much time do I have left. Okay. 10 minutes, okay. So I'll try to quickly tell a little bit, you know, what else we'll be doing in my lab. And we are interested in looking at structures of viral capsids. This is something I was talking also yesterday about this at the conference. So the capsid expands during DNA translocation into the capsid when virus particle assembles, and we determined structures of unexpanded and expanded state of the capsids. This work was done by Oliver Bayfield, and showed that during this process, each subunit of the capsid, and there are 415 identical subunits, each subunit undergoes conformational changes, very significant conformational changes, which allow capsid expansion. And this process, you know, doesn't really result in breakage of the capsid and leakage of the DNA. So it's quite amazing mechanism. And we are interested also in structures of complete viruses, and Oliver recently determined structure of cross-phage. So this is virus. These are viruses which are most abundant in human guts, and there is a lot of hope now that they can be reprogrammed in future to be used as personalized pharmacies for production of proteins and peptides, which would be beneficial for our drugs. And what was really unique in this case was that we observed parts of the same protein inside the viral tail. Actually, no one seen before proteins in viral tails. And we can even identify what protein was stored in viral tail from the density Oliver generated. And another domain of the same protein was actually inside the capsid. So this protein would be translocated first, and it will form transmembrane tunnel. And protein also carries RNA polymerase subunits, which we think will be translocated next into the host cell during host cell infection. And then this will be followed by translocating viral genome. So apart from bacterial viruses, we started working on archaeal viruses. So some of them look similar to bacterial viruses, some not. So this is work which is done by Julia. And we have a bunch of other bacterial viruses which we are interested in. So this is one of the giant viruses which was provided to us by a collaborator. And so far, research efforts in my lab and in several other labs have actually concentrated, focused on this group of viruses, which contains, which is comprised of tail bacteriophages and also herpes viruses. But there is another big group of DNA-stranded, double-stranded DNA viruses, which contains bacterial tectiviruses and also a number of eukaryotic viruses, including pox viruses, which are human pathogens. So we started working on these viruses, and Brianna, a very talented PhD student who joined from US, he already obtained medium resolution crying structures for two such viruses, YKO, which is thermostable virus, and PRD1. We are trying to resolve structures of a unique vertex through which these capsids are filled by DNA. And these viruses use completely different machinery for DNA translocation. So instead of terminase protein, they use so-called FTSA like DNA translocases. And Paul Bardi, so another very talented fellow, actually has his own postdoctoral fellowship now. So he's establishing technology at York for cryo-electron tomography. And here is a section of whole cell. Actually, it's not a section, it's a whole cell. So Pavel managed to find conditions where he can grow very, very thin cells. And having a thin sample is very important for performing electron tomography because electrons are absorbed very easily by matter, so you need to have a thin sample. And he recorded this tomogram where you can actually see viral particles during the interaction with the host cell. And the idea is to derive accurate structure for such viruses, and maybe also try to derive structures for viruses which are actually assembling in the host. So it's something we want to do in future. Yes, so we have a bunch of other projects work on helicases, and this project is done again in collaboration with Oxford Nanopore Technology. So as I say, they currently use technology where they have two separate proteins, a pore and the translocase, which drives DNA at a constant speed through the pore. And what they really want to do is to create a single protein which will have both functions in the same polypeptide chain. So it will serve as a pore and it will serve as a machine which also at the same time translates single stranded nucleic acids. So we determine structures through helicase, cell, and complex RNA. And we are now extending it with transmembrane segments to see if we can actually dock it to the lipid bilayer. Okay, just a final example of work we're doing. We also started a project on applying microelectron diffraction at York. So this is a technique which was developed in Tamar Bonnet lab initially, some maybe 10 years ago. And the hope was that you can actually determine structures of proteins using very, very tiny crystals. Right, but I think with proteins, because electrons interact with the matter so strongly, there are a lot of secondary scattering events. So it's really difficult to deconvolute data, and at the moment, this technique is really limited to determining structures of relatively small molecules, and we determine structures for several organometallic compounds, which because we're in chemistry department, there are lots of chemists who produce synthesize novel chemical molecules. And in many cases, they can only obtain very tiny crystals, which are, believe it or not large enough to determine structure by crystallography. What you can do, so you can just scatter some of these crystals on electron electron microscopy grids, sticking to the microscope. And what we often see is that there are tiny crystals, some of them even belong to different crystallographic symmetries. And you can, by rotating your crystal, you can collect electron diffraction data set in a very similar way we collect data from protein crystals and then derive structure for such compounds. And this technique is particularly suitable for compounds which have very, very short time length because you can freeze them and you can determine structure under cryo conditions. Okay, so I think that's everything I wanted to tell you about. I'd just like to thank current group members, former group members, including Leong and Feng, collaborators, really a large number of collaborators over the years. And yeah, thank you very much.
Thank you, Fred, for the very interesting historical background of how you came to today, and also the insight of the sharing on virus work, particularly on the protein, viral protein that binding to the nucleic acid, and also the packaging. So I would like to open the floor for Q&A. So we would have 10 minutes for this section. So take your opportunity, and we have Fred here to answer your questions.
Don't be shy.
Yeah, don't be shy. Yes, please. Carol.
I want to ask, is that the helicase and the DNA transport case, they share the same mechanism?
We have somewhat similar mechanisms, so kind of in looking at the big picture, it's a similar mechanism of translocating DNA. So in the case of helicase unlike with double-stranded DNA, so they translocate single strand of DNA. Whereas double-stranded DNA translocated by a double-stranded DNA, kind of more rigid. But the mechanism is quite similar. There was very nice work published with Jan Lorla from LMB. They know Jan. It was published 3-4 years ago. It was basically published PNAS on FTSK, like hexameric assembly of FTSK APKs. And the mechanism is again the same. So there are conformational changes running around the frame. Okay, and ATP hydrolysis happens simultaneously in 3 subunits, and 1 subunit ATP is found. The next subunit will be transition state already formed, and the next subunit phosphate detaches from the gamma phosphate of ATP. And it is actually, I think, this dosage of the gamma phosphate which generates the working stroke. So that's when largest conformation changes happen. But the overall mechanism is quite simple.
So the structure biology has evolved on the time of John Cantrell or Max Time, they calculate point by point manual data collection to a highly automated and computerized workflow. So in a sense, it's a good combine in physics and mathematics still necessary in structural biology. But, yeah.
Yes, of course. I mean, maths are still very important. And in particular, in structural biology is now moving very much into cryo. So if you have anything bigger than 100 kilodalton, it's really considered doing cryo. Because we will learn more from the structure. But the big problem with cryo is that in your sample you will immediately see, in most cases, there will be different conformational states present in your sample. And there are lots of improvements which can be done using the automatic assignment of these different states into different classes. And I think there is a lot actually code for machine learning, for example. But it's a very fast developing area, and there's a lot of maths involved in calculation. So you're right. I mean, now you essentially press the button and get your structure, but after you press button, there is a lot of Fourier analysis, correlated function, and yeah, now we have so powerful computers we can do it a lot faster than before. But still, some tasks take sometimes have to wait for half a day, to get your calculations done. You want supercomputers. So I guess maths is still very important. And even to process your cryo data, I think maths might go back on. It always helps.
Thanks, Fred. Yes, Vicky.
So it is interesting that you're developing new routines for the UNE. So I work with hospital for sequencing before. So like my struggle was I had to do everything USD by not introducing any bubble. So I mean I know by using like this single protein. So is it possible for it not to be that sensitive when the bubbles was actually introduced to the ball?
Well, I guess you better talk to Oxford, but you know why you can get bubbles forming, but I suppose maybe something about simple, maybe, you know, sorry, if there is any detergent, you know, usually mooring it may help, also might not help solubility. Yeah, I know that you know everything is kind of set up, right? That we don't even know what is chemistry of membrane by way. But we can do mass spec and find out, but you know, this sort of don't tell anyone. Even us, you know, even though we collaborate with somebody else, you know, how this is my time. No. But yeah, I think you'll better talk to. But they are really you know driving developments in this area, and what they do, the UK, they work with probably at least 10 different universities, you know, including ours. And whenever there's you know someone working with some nucleic acid cores, they usually connect to them, and they fund PhD students. So they funded 2 PhD studentships in my lab, which is great. You know, you know, when you get funding, right, you just do things, right, because it's how it works. Research. You know, funding is not easy to get.
Yeah, right. Thanks, Brad. I think we are there any question in FP? Maybe we take one from there, and then we should end our seminar this morning. And I say 10 minutes or something. Sorry, and we have 5 more minutes. Or in mind. Brett, there's a question here. Does ATP or ATP have a direct structural role in the formation of nuclear capsid from addition?
Oh, it's a good question. So as far as I know, ATP has no such role. But when you know RNA sequence is read out, you know, once a copy of the RNA will be synthesized during viral production, there is a mechanism which actually breaks up this nucleic acid complex, and you know during which RNA becomes accessible, and this process of synthesizing copy of RNA will definitely you know require ATP. It will be somewhat protein. But formation of the nucleic acid complex is presumably just driven by you know interactions of the protein, proteinase, protein, and RNA.
All right, I hope perhaps address additional question. We have 3 more minutes. Let me take advantage of asking one question about the Nipah virus that you show. You mentioned about there's 3 bases in and 3 bases out, 3 bases in and 3 bases out. And you also mentioned that it's non-specific binding. So in this case, what's the mechanism of determining the 3 bases should be in and out?
Yeah, it's a good question. But interaction involves essentially interaction with the sugar phosphate backbone of RNA. And where the subunits binding site is exposed, then you know C nucleotides would be nucleotides of the C consecutive bases of the C consecutive nucleotides would be bound. So this binding growth. But then there is spacing between adjacent subunits, so that's where bases of CERs are essentially exposed to the solvent.
Without the selection.
So wherever there is binding, binding site, it's just bind 3 nucleotides. Yes, all the 3 nucleotides are not, largely instructions.
So I think we have one more question here.
Sorry?
There was a recognition that
Theories thought about it, so this it's possible, we never done it. But in principle, yeah, you can try to reprogram it and maybe use such properties for some part of application. For recognition of specific.
One last question here from FB. Hi, Prof. Brent, thank you for the interesting talk. I have one question about the DNA helicase. Is there any structural difference in the DNA helicase that bind to double-stranded DNA and single-stranded DNA viruses?
So yes, so the helicases belong to different classes, and actually, this question probably about classification of ATPase, not just helicases. But helicases normally translocate single-stranded DNA, and there are 6 different types of helicases. Some of them are monomeric, dimeric, several are hexameric. But the DNA translocation motors, which translocate double-stranded DNA, so there is more variation between them. So the motor, which is viruses which are where we have this terminase protein, which has ATPase activities, they form pentameric assemblies with higher sequence similarity to actually monomeric DNA translocase, which were again both-range mechanisms. Then there are FTSK-like translocases, which have somewhat different forms and belong to different family of ATPases. And the second large group of double-stranded DNA viruses, for example, they use FTSK-like translocases. It's kind of known because they sequence similarity to FTSK translocases. But curiously enough, the structure of the FTSK motor, which was determined by LNB, by Ian, it's a hexameric. It's a hexameric ATPase motor, and many of them are hexameric. But when lower resolution data were obtained for these double-stranded DNA viruses in Varchula, double-stranded DNA viruses which use FTSK-like translocases. It's really bizarre, but there is clearly there is a 12-stranded assembly, and you kind of think, normally FTSK translocases take 6 strandeds. In this case, 12. Why? No one knows. This is something interesting to resolve in future.
All right. Thank you, Fred, for all the sharing. Before we end, I would like to invite our Deputy Director of INBIOSIS, the goal, come to the front and some token of appreciation for Professor Fred.
Oh, there.
That's okay to get doing for you.
Thank you, Dr. Goh. Once again, on behalf of Institute of Systems Biology and Malaysian Society for Biochemistry and Molecular Biology, and also our research structural biology research group in UKN, we would like to thank Professor Fred Antson again for this very insightful seminar. Thank you, everyone, and thanks for all the participants joining in the FP. We would like to have a group photo section as well, right?